All right, let's take a tour of the prokaryotic cell. And what we're going to do first is talk about all the things that are inside a prokaryotic cell. And the good news is there's not that much there. These are pretty simple. So it won't take that long to go through them, but they're important. It's simple, but a very functional cell. So everything that's in there is important. Let's start by talking about the genetic material, which is the very inside. So when we talk about prokaryotic genetics, we refer to the nucleoid, which is the region where the prokaryote's genome, the DNA, is located. And in prokaryotes, the genome is a singular circular chromosome. And contrast this to your cells, which have 23 pairs of linear chromosomes. Again, in a prokaryotic cell, it's a single circular chromosome. And it's anchored to the plasma membrane. So it's not surrounded by a nuclear membrane like a eukaryotic cell, but it is anchored to the plasma membrane. So the genome is a single circular chromosome. It's located in the nucleoid. Now, many bacteria have little extra pieces of genetic material called plasmids. These are also circular. They may contain anywhere between 5 and 100 genes, and they're not critical typically to the everyday function of the cells, but they may give that cell extra properties such as antibiotic resistance, for example. Let's look at some pictures here. So here is we're just going to look at the interior right now. Here is the genetic material. So this region where it's located would be the nucleoid. And we're just looking at the same thing here. This is the genetic material, and this is a transmission electron microscope image of a bacterial cell. So it's pretty cool that you can see in an actual photograph of a bacteria, you can see this nucleoid region that contains the DNA. Now, if this cell had plasmids, it would have little extra circles which aren't shown here. We will be working with plasmid DNA in the laboratory when we look at pGlow later on in the semester. And so you'll have a better sense of how plasmids are different than the main genetic material. But just remember, they're just little extra pieces, and bacteria actually have the capability of sharing plasmid DNA with each other in a process called lateral gene transfer. Something else just to kind of keep in the back of your head that we'll talk more about later on. Oops, I went too far. There we go. All right, so now we talked about the nucleoid. Again, we got another picture with kind of the same thing here. And then that nucleoid, that genetic material, is actually located in the cytoplasm. And the cytoplasm has water as a solvent. So we talked about how things dissolve in water. And you want to think of the cytoplasm as having so much stuff dissolved in it that it's almost a gel. Maybe the consistency of Jell-O. We used to think of it as this really fluid mixture, but actually it's so packed with materials that it's really a much thicker solution. It's got enzymes, it's got nutrients, it's got waste the cell is getting rid of, and it has some dissolved gases. It's also, in addition to the nucleoid, which is again the genetic that contains the genetic material, it contains organelles called ribosomes and structures called inclusions. And so those are next on our to-do list here. There are a variety of different kinds of inclusions in bacterial cells. They basically function in different ways. So some of them are reserves. They can hold polysaccharides. They might contain lipids or sulfur. In some bacteria, they actually contain gas, and they can allow the bacteria to control how buoyant they are in a solution, or if these bacteria are living in a pond, for example. Some inclusions are called magnetosomes, and these actually have magnetic particles that, again, the cell uses to control their movement in three-dimensional space, where they're located, based on their magnetic properties. And then there are also inclusions called carboxysomes, which are found in photosynthetic bacteria, and they just contain enzymes that are critical to carbon fixation in the photosynthetic process. So inclusions they're basically a structure that many bacteria have. They have fulfilled different roles, and they have their storage, they're specialized, they have their movement depending on the bacteria and the structure. Also inside the bacteria are ribosomes. And so bacteria have ribosomes just like you do. These are not surrounded by by membranes in either eukaryotic or prokaryotic cells. And these are the sites of protein synthesis. They're found in prokaryotic cells either in the cytoplasm or attached to the plasma membrane. And just quickly to contrast that with your cells, you have ribosomes in the cytoplasm of your cells or attached to the rough endoplasmic reticulum, which is a membrane that's part of the intermembrane system of eukaryotic cells. Prokaryotic cells, these are small. Prokaryotic cells contain tens of thousands of ribosomes. So there's a lot of ribosomes in a prokaryotic cell. And prokaryotic cells are a complete ribosome is got made up of two smaller subunits. So we have a small 30S subunit and a larger 50S subunit. And when they come together, they form something called a 70S subunit. And of course, or a 70S ribosome. And of course, no one likes this because 50 plus 30 isn't 70. And so I always take a moment to explain that the measurement S refers to Svedberg unit. And Svedberg unit is a reference to how fast the particle sediments in an ultracentrifuge. It's basically an ultra high speed centrifuge. And when the 30S and 50S both sediment based on their density, as does the 70S complete ribosome, and it just so happens to sediment at 70 Svedberg units if it's complete. Anyway, not critical that you memorize that, but hopefully it explains why 50 and 30 makes up a 70S ribosome. One more comment about ribosomes while we're on this slide, and that is that you have 80S ribosomes in your cells. Bacterial ribosomes have the same function as eukaryotic ribosomes, but they are different enough that many antibiotics that you're familiar with actually target ribosome function in bacteria. So they'll prevent bacterial ribosomes from functioning, but they won't damage your ribosomes, which makes them a good therapeutic treatment for bacteria. So a common antibiotic that you might have heard of that targets ribosomes would be tetracycline. Another one would be erythromycin. All right, so now we've talked about the major parts of a prokaryotic cell that are inside, and our next lecture snippet is going to be about prokaryotic cell membranes and their function, their structure, and their function and their role in the cell.